I don't think that we should see real-world data analysis and randomized controlled trial data analysis as two different things, two opposite things. They must be seen as complementary.
But what then becomes, to me, the next layer that is really interesting is to look at these conditional associations, conditional effectiveness of therapies, where we have these subsets of patients and we start talking about in whom it pays off, at least from the point of view of effectiveness, to use treatment A rather than treatment B.
This will help to compare results across studies and also to have a guarantee to do all the same thing, to be all on the same page.
The problem is that if there was a silver bullet, we would have already found it. We were all hopeful that NFL, for example, is going to be it, and we're now learning that this is all conditional. In some populations, these predictors may work, in other populations, they may not work.
Hello, everybody. My name is Maria Pia Sormani. I'm a professor of Biostatistics at the University of Genoa in Italy.
Hi, my name is Tomas Kalincik. I'm a neurologist and applied statistician. I lead the Neuroimmunology Centre and the Clinical Outcomes Research Centre at the Royal Melbourne Hospital and University of Melbourne in Australia.
And I'm Brett Drummond. You're listening to the ECTRIMS podcast, the official podcast from the European Committee for Treatment and Research in Multiple Sclerosis. One of the most transformative changes in MS research over the past decade has been the establishment and growth of MS registries around the world. While this has provided researchers with a vast quantity of clinical data collected over long periods of time, it has opened up important discussions around how best to utilise this information to answer critical questions around the treatment of people living with MS and the potential to shift towards a personalised medicine approach. My guests in this episode, Maria Pia and Tomas, are experts in the field of using real-world data to provide answers to key questions in the treatment of MS, having published extensively in prominent journals across a long time period. Maria Pia, Tomas, thanks for joining me.
Thank you for having us.
Thank you.
So I guess to briefly start this episode, it's a discussion we always have when we discuss the use of this data. I was wondering if you could just talk to us briefly around the comparison of using clinical trial data versus using real-world evidence.
Well, I don't think that we should see real-world data analysis and randomized controlled trial data analysis as two different things, two opposite things. They must be seen as complementary instruments we have to answer different research questions. So, of course, the randomized controlled trials is the gold standard for assessing the efficacy of a new drug. So we need a controlled experiment to understand whether the drug is working. Then the real data, observational studies are needed to complement this kind of information. I don't know, Tomas, if you agree on that.
Yeah, absolutely. The data are generated with a purpose in mind. And I'd like to highlight that observational data are actually more prevalent than people think. It is something that we work with as clinicians every day. But also randomized controlled trials become observational data once they are used outside of the scope of the primary research question of the original trial. And you, Maria Pia, and your team have an extensive experience and track record in repurposing data from randomized controlled trials to answer new questions.
Yeah, that's correct. Because, I think that randomized controlled trials require a huge amount of money, so they are a great investment. And the sponsors usually are just interested in the primary research question. So is the drug working, is it effective or not? But in the end, in the randomized controlled trial data, there's a lot of information that can be extracted post-hoc. So this is something that we have been doing in the past, establishing correlation between different outcomes. Also to understand whether, for example, MRI outcomes are able to predict clinical outcomes or the treatment effect on clinical outcomes. So you're right, randomized controlled trials can be treated as observational studies post-hoc.
Yes, the methodology of the observational data and the analysis is specific, and it is more complicated than in randomized controlled trials, mainly because observational data violate the assumption of randomness of treatment allocation, at least for the purpose of the specific research question. In observational data, such as registry data and cohorts, we have been using statistical methodologies to emulate clinical trials, to simulate randomization, whether with propensity score approaches or more advanced approaches in counterfactual framework, in causal inference framework. I would like to ask you, with your personal experience with using this data, and perhaps you may use some of the examples of your own work, what is the approach that you need to take to answer these questions? And I know it's a broad question because different questions have a different requirements.
Yeah, of course. But, there are many different statistical techniques to adjust for the lack of randomization we have in observational studies. But, as you may know, I've been always a bit skeptical about using observational data for answering comparative effectiveness questions. I think that observational data are very useful to address all the things that are missing in a randomized clinical trial. So, first of all, the long-term safety, the usability, how patients perceive treatment. And I mean, in the observational studies world, there have been many examples of very useful information that have had a clinical application. For example, all we know about PML in people treated with natalizumab comes from the observational world. The extended dosing interval as compared to the standard dosing interval that reduce the risk of PML comes from very large, well-designed observational studies. So another field that I think is very important is all the answer we have about pregnancy. Of course, we can't address pregnancy in a randomized control studies. My criticism it's a criticism that I give to myself also, because all of us has been tempted to force a little bit the idea of comparing different drugs to find a sort of ranking. And this is something that, of course, pharma companies like a lot, but I don't think that this is what have real clinical implications. So we know that the drugs are in class of efficacy, so we have very potent drug, we have less effective drugs, but I don't think that in the end the clinician choose a drug according to its ability to reduce relapses by 30% versus 35%. So, the choice of a drug is based on different aspects that, of course, are clarified by observational studies.
Yes. I think that whole field and whole effort has evolved. When we first started comparing therapies head to head, we were genuinely interested in exploring in general principles, broad brushstrokes. How does the treatment A compare to treatment B overall in its capacity to reduce relapses, prevent disability worsening, stimulate disability improvement even. But that is only very basic information and very crude information, as you say. That is actually it's important to have that knowledge, that understanding, in my view. And we still do that because we know that companies will not come together to compare treatment A to treatment B. Once a phase 3 trial is done, it is done. Yeah. And I think it is our role to approach the additional questions of comparison of treatment effectiveness with the data that are available either through registries or from randomized controlled trials if we use them in a smart way. But what then becomes, to me, the next layer that is really interesting is to look at these conditional associations, conditional effectiveness of therapies, where we have these subsets of patients and we start talking about in whom it pays off, at least from the point of view of effectiveness, to use treatment A rather than treatment B. And this can be approached through many different approaches, stratifying the large data sets into subsets of people in certain clinical situations, such as people who just recently had a relapse or clinically stable cohorts, or approaching in a slightly more continuous way, taking data from multiple trials, converging them together, and using some more sophisticated instruments to calculating or estimating a treatment response score in comparison of two different therapies. And again, I think you have some interesting examples of that.
Yeah, I fully agree with that. We know that the treatment effect is always expressed at the large group level. So reduction of relapses of 30% does not mean that every patient has a cut of 30% in their relapse risk. There are responders and non-responders. So this is the big question we have to answer, having many drugs available now. And of course, observational data can help a lot in answering this question. And also, I think that for the future, we need also to explore questions that are not explored by pharma companies, like association of drugs with exercise, diet, and other approaches to treat MS to improve the quality of life of MS patients.
Yeah, absolutely. I mean, the magnitudes of treatment effects that you mentioned, they in large cohorts, of course, what we detect can be tiny, negligible. Purely from a methodological point of view, and I think there is an important distinction there between methodological and clinical point of view. To me, it is important to understand with a high level of certainty that there is a difference that is of a small magnitude, because that allows us to tell the clinicians or tell ourselves that if you treat with treatment A and treatment B, yes, there is a difference of, let's say, one relapse every 20 patient years. And we can arrive at that difference with a good statistical certainty, so with a little doubt. But importantly, then the clinical translation of that information is, does it really matter?
Yeah. But I fully agree with you. But, as we discussed many times, the risk is that for clinicians sometimes take a very binary message. The comparison is positive, one drug works better than the other. So I agree with you. It is very important also to understand the size of the effect that a study is analyzing, because with observational data and such large cohort, we can have significant results also with very, very small and non-clinically significant difference. So the risk is for clinicians to be very careful in evaluating the results of a study where, for a clinical trial is different, because if the p-value is significant, there's a story behind. The study has been designed to reach that result. So if the p-value is significant, this means that the difference is clinically relevant. This is not the same for observational studies, where usually we analyze all we have, because it's stupid to discharge information if it's available. So our aim should be to clarify how to interpret the results.
Yeah, I fully agree. And that's actually one of the reasons that in the last few years we've been really trying to minimize the use of p-values in non-trial designs, because p-values from the origin come from randomized trials. And rather than reporting p-values, we focus on reporting the point and interval estimates of the magnitudes of difference, of course. But I think that is a really important point, that as authors and editors of journals, it's our responsibility to explain, to provide that editorial content in our discussions, in our conclusions, our communications, to give the clinicians a sense of perspective that, albeit our study shows a tiny difference between two therapies that have similar mechanism of action, that the question is, does this tiny difference translate into a difference in clinical decision making, or on the contrary, does it provide the clinician with reassurance that if they choose one or the other therapy, they're always going to choose a reasonably safe, from the effectiveness point of view, approach to therapy?
Fully agree with that.
So I wonder if I can jump in, because it would be remiss of me, with the two of you sitting in the room, not to ask you this question. Obviously, with the data that you're using at the moment and the sort of questions that you're asking and the ability to shift to use this towards coming up with things like prediction algorithms and understanding potential outcomes in individual patients. How do you feel around whether this will start to be used routinely in clinic? Do you get the sense that there will both be buy-in from neurologists and clinicians to use this information to make these decisions? What do you think are potential barriers in that? What are timeframes? It's a big question, but if anyone's going to be able to answer it, it's the people sitting with me here.
Yeah. It's very challenging to answer, because the issue is with MS. For tumors, it's easier to do a personalized approach, because you have biomarkers, you have the tumor profile, you have a tissue, you examine the tissue, you have a genetic markers of the tissue. MS is such a heterogeneous disease, and we have been struggling for years trying to find prognostic factors, markers of response, but we really don't have something strong. We have some lines, but demographic information, males versus females, something like that, but we really don't have profiles of patients that are responding to one drug or the other. So this is the real problem. So we're trying to refine our algorithms, but up to now, we still don't have something strong.
I think that there is a methodological aspect to the answer, And that leads us to understanding that the group level difference doesn't equate an individual difference. And there are pieces in general literature, and we have recently published a couple of reviews in MS literature in Brain and in Gen and P on discussing how to transition from the group-based difference to an individual difference. So that's a methodological aspect of the work. But then there is also the practical aspect of the work. If we calculate a score or generate a score, albeit nicely validated and well-fitting, with, let's say, a machine learning algorithm that uses high degree interactions among individual patient information, and then we tell the clinician, use this in clinical practice, clinicians are not going to use that because that requires a clinician going online somewhere, putting the information in the machine and asking the black box to spit out a piece of data that tells them this individual will be such and such. What works well with clinicians is being able to do on a piece of paper or in their head simple procedural classification of patients. It works well in stroke, and it has worked to some extent in MS when they're able to say, okay, this is a young patient who has a relapse and therefore a large lesion on the MRI, and therefore they should be on a high efficacy therapy. So I think we need to be aiming at trying to simplify that information in terms of prognostic markers. The problem is that if there was a silver bullet, we would have already found it. We were all hopeful that NFL, for example, is going to be it, and we're now learning that this is all conditional. In some populations, these predictors may work. In other populations, they may not work. So I think the task is still out there for us to find a way how to combine these different predictors in subsetting patients into these categories that would be accessible to the clinical user.
Yeah.
So focusing back on the use of real-world evidence to start with, what would you say are some of the biggest success stories over the past couple of years in terms of answers that we've been able to get really clear, questions we've been able to get really clear answers to using this information?
Well, for me, one of the biggest successes I mentioned before was the PML story. I mean, everything we know about PML, I think that neurologists are very able now to manage the risk, to minimize it, to treat patients with natalizumab. That is a very potent drug. And we learned a lot. So this is a big success coming from observational data.
To me, it's how our gradual step-by-step approach to comparing therapies has led to shift in thinking around using therapies. And as I said earlier, this comes in layers from simpler and broader questions, less specific questions, to more specific questions about subpopulations. And if we compare where we are now compared to where we were 15 years ago, now most of the clinical world says we got to treat with high efficacy therapies and we have to start them early. Yeah. Whereas 15 years ago, the debate was out there. Yeah. So there was this gradual accumulation of evidence has led to a shift in mindset. And part of that was comparing the therapies in a way the results were really intuitive, in keeping what a clinician would expect to see if there was a clinical trial scoped around the question of comparing a treatment A to treatment B in a specific clinical setting. Now we are entering into an era where I think the observational data are providing us with a powerful instrument to really unify the way that we approach studies, not only in observational realm, but interestingly, paradoxically, observational data have now the chance to inform the design of clinical trials, specifically of their outcomes. Maria Pia here has recently published in Multiple Sclerosis Journal a study that used a package that unifies a definition of confirmed disability worsening. Our team, CORE, has designed its own package for confirmed disability progression. And I think it's really exciting to see that we can offer these packages to analysts around the world.
Yeah, because, one thing that we have maybe underestimated is how we calculate disability progression, for example. So we have been criticizing EDSS as a bad scale for years. But, we put a lot of attention on how the event of progression has been estimated. If you go around across all the clinical trials, you see that, the definition is broadly the same, but there are a lot of details that made the definition different. And we learned this having access to clinical trial data, so having the possibility to recalculate disability progression. We learned that, it was really difficult to reproduce the results of every single trial because the details are so many that it's impossible to to have them all perfectly reproduced. Also because these small details are not usually reported in the statistical analysis plan or in the protocol. So we decided to run this initiative that is a pretty trivial thing, that to have a unified approach to the calculation of confirmed disability progression. This will help to compare results across studies and also to have a guarantee to do all the same thing, to be all on the same page. And of course, this was a result of our continuous analysis of observational data, because we had to face a lot of problems, with the observations that are not equally spaced in time. So we are very well aware of all the problems we have simply calculating confirmed disability progression. Mm-hmm. So I hope that this will be a big work putting together people from pharma company and registries.
Yeah, and I think in terms of this, the sorts of questions that we can ask using this data are probably endless. Having had many conversations with Thomas in the past, I know he doesn't struggle with coming up with questions that he wants to answer using this data. But what limitations still exist in terms of being able to do that, Maria Pia? I know before we started recording, you were talking about data collection as being part of that.
Yeah. At least I know the situation in Italy. Of course, it would be fantastic to have a unique way of collecting data across registries, but also, I'm talking about Italy, so across the same country. I see that in Italy, many neurologists to participate in the registry, in the Italian registry, also, but also to MS-base, have to put the data in a different system, so to reinsert the data. And for them is a big burden to, to insert the data into the hospital system and then to insert again the data in a different system. So I know that in other countries the situation is different, but of course these technical things must be solved to have a better data quality.
Mm-hmm. That linkage may be possible through, again, technological solutions. We have recently seen efforts in linking the electronic medical record systems in the UK with databases. And I think that is potentially a way for us to approach this. If we can offer clinicians systems whereby they only enter the data once, and with a little gadget are making sound simple, it is not simple at all, then have those data curated and transferred in an appropriate format to another data repository. That would, of course, take that extra work out of the process and also minimize the risk for erroneous data entry.
But with artificial intelligence methodology, for example, we can think to systems that put the data extracting from what the neurologist tell to the patient. So a voice system that put the data into a database, so the neurologist is aware that he must put some data into the system, but he does not need to write. Just asking the patients and collecting the data they can be. Of course, this is wishful thinking.
Well, it may be closer to reality than we think, because there are already systems that listen to the conversation in a clinical appointment and draft letter notes for the clinician. Yeah. So I think that's a pretty the science fiction is becoming reality.
Yeah.
So continuing on thinking into the future, what research avenues are you most excited about in the next sort of 12 months? What do you think in this space is going to be some of the big developments?
You mean in MS, of course.
In MS, yes.
I'm very excited about all the research about EBV virus. I think it's promising, even if it's very challenging, because test the hypothesis. Also, trying to assess the efficacy of vaccine against EBV virus is extremely challenging. Mm. But I think this is an exciting field of exploration.
Yeah, I'm on the same page, Maria. I think that to me, the next exciting step is bringing the realm of the world of data together with the world of biology. Mm-hmm. And the tissue-based, the blood-based research, the in-depth understanding of the immune system, and to see how what we see on the surface, that is in the clinical observations or in radiological findings in these patients, how does that correspond to underpinning biology of MS, but also the other conditions? Today we are at the extremes pre-day, and there has been a lot of fascinating data presented about the biology that differs vastly across the different neuroimmunological conditions. And being able to link the behavior of the disease at an individual patient level with that biological process, not only generally defining the disease, but also specific to the concrete patient, is a very exciting, to me, very exciting linkage. And why I am particularly excited about it is that with sufficient understanding of that association, the clinician will in the future be able to say, because the person's presentation is such and such, the underpinning biology is most likely to be this, and therefore a treatment that approaches that particular biological pathway is the most likely treatment to help this person.
I mean, exciting things to come. I know we could continue talking about this for a very long time, but I think we will finish on that note. And thank you both, Thomas and Maria Pia, for your time today and sharing all of those exciting insights with our listeners.
Thank you very much.
Thank you.
And thank you all for listening. Once again, I'm Brett Drummond, host of this podcast episode and co-founder of MStranslate, an independent resource centre that aims to simplify the complex wealth of information about MS for the greater MS community. The ECTRIMS podcast is produced and hosted in collaboration with two key partners, MStranslate and the MS Journal, a peer-reviewed international journal that focuses on MS, neuromyelitis optica, and other related autoimmune diseases of the central nervous system. The 2024-25 ECTRIMS podcast series is supported by educational grants from Alexian AstraZeneca Rare Disease and other providers. Educational grant providers have no input in the podcast content. You can read more about the topic of this latest podcast episode and more on the ECTRIMS website, ectrims.eu. Do subscribe to the ECTRIMS podcast on your favourite podcast channels and follow ECTRIMS on X, formerly Twitter, LinkedIn, Facebook and YouTube.